Okay, so welcome to this next video in which we're discussing the adaptive immune system and immunosuppressants. Okay, so we've discussed that in the humoral adaptive immune response, professional antigen-presenting cells such as dendritic cells and macrophages at the site of infection are going to phagocytose our extracellular pathogen, because remember we're talking about humoral or extracellular response at the moment, then they're going to break this pathogen down, and they're going to break down surface antigens on the surface of the pathogen, and they're then going to put fragments of this antigen on the surface, on their cell surface membranes, where it's bound to the major histocompatibility complex class 2. These professional antigen-presenting cells are then going to go in lymph vessels to lymph nodes, and they're then going to be exposed to a huge number of T lymphocytes. Now, what they need to do is they need to find a CD4 positive naive T cell which has a perfect T cell receptor, a T cell receptor that is made for this antigen fragment that the professional antigen presenting cell is now presenting. Okay, so it will have to search for quite a while, because you know there are a lot of CD4 positive naive T lymphocytes, and not all of them will have T cell receptors that are going to bind to your antigen, so it needs to search and search and search for a T cell which is a match. But let's now talk about what happens when it finds a CD4 positive naive T cell that is a match. Okay, so let's put our T lymphocyte here, and I'm going to elongate it a bit. The reason is that I've got a lot to sort of fit in here to show the interaction between the two. Okay, so here's the nucleus of the naive CD4 positive T lymphocyte here. Okay, and now it's got this professional antigen presenting cell, which I'm just going to draw as a blob, and the reason is that it's far easier to show it if you draw it as a blob. But it will most likely be a dendritic cell, so it will have that silly structure where you have dendrites all over the place, but that's impossible to show what we need to show with. So we're drawing a blob, so you could think of it potentially as a macrophage. Okay, so here's its nucleus. Okay, right. So this professional antigen-presenting cell has got the antigen fragment on the surface of MHC class 2. Okay, so here's the antigen fragment here, okay, and it's bound to the major histocompatibility complex class 2. So the antigen fragment is there in blue. We'll have MHC class 2 in green here, okay, and it's now presenting this antigen fragment to this T cell, and let's say this CD4 positive T cell has a complementary receptor, has a complementary T cell receptor, and the T cell receptor is going to have grown somewhat just to fit everything in. Okay, so there is our T cell receptor. So let me label things up. So this is major histocompatibility complex class 2, or MHC class 2. This here is our T cell receptor, or our TCR for short. I'm going to make that antigen fragment a little bit bigger now so that it fits into that gap in the T cell receptor that I've drawn. And I'll draw the T cell receptor in pink here, so I'll colour this in pink. Okay, there we go. That's the T cell receptor. Now, why was it so important that this was a CD4 positive naive T lymphocyte? Well, it's because CD4 is now going to bind to MHC class 2. So let's let this be the CD4 molecule here. Okay, so I'll colour that in orange. Now, oh actually we did it in pink before, but I can't draw it in pink as well. I'm going to draw it in orange, even though that's dangerous because I coloured CD8 in orange before, but this is a CD4 molecule. Okay, CD4 will bind to MHC class 2. CD8 will bind to MHC class 1. So these ones are not going to react at all to these antigen presenting cells that are presenting it, the antigen fragment on MHC class 2, simply because their CD8 will not bind to the MHC class 2. So you need not only the T cell receptor to bind to the antigen fragment on the MHC class 2, but you also need this CD4 to bind to the MHC class 2. And when you've got both of these things, that delivers what's known as signal 1 activation to the T cell. Okay, and we'll come back to the actual ins and outs of signal 1 later, because we're going to talk about the immunosuppressants, and these will involve actually looking at what's going on inside the T cells, because a lot of the immunosuppressants are going to work by stopping the activation of T cells and thereby cutting off all of the adaptive immune responses. Okay, but we'll come back to that later. Okay, so this signal is signal 1. Okay, now that is not enough to activate this CD4 positive naive T cell, and this is where we're coming back to this discussion of what it means to be a professional antigen presenting cell. In order to activate this naive CD4 positive T cell, you need to give it signal 1, and you also need to give it signal 2. And to give it signal 2, you have to put what are known as co-stimulatory molecules on your surface, and there are two main co-stimulatory molecules. Okay, so I'll draw these here. Now, what causes this professional antigen presenting cell to put co-stimulatory molecules? And that's a key word, or a key term, so I'll put it over here. Co-stimulatory molecules. Well, basically, this antigen presenting cell, which is a dendritic cell or a macrophage, will also have what are known as pattern recognition receptors on its surface. So let's put one of these here. So let's say this is a pattern recognition receptor. And these pattern recognition receptors bind to basic molecules which are associated with pathogens, basically. Okay, so if you think about it, how can you recognize a cell as being a pathogen rather than a human cell? Well, basically, you're going to have to look for molecules that that pathogen has, which no human cell would ever have. Okay, so these sort of molecules that pathogens have, which human cells would never ever have, are what are known as pathogen-associated molecular patterns. Okay, and these don't need to be proteins. Antigens were proteins. Pathogen-associated molecular patterns are generally things like double-stranded RNA, things like that. Sugars, polysaccharides that we would never ever have, lipid molecules that we would never ever have on a human cell. Okay, so these are not necessarily proteins at all. So pathogen-associated molecular patterns, and these pathogen-associated molecular patterns are a giveaway sign that this cell is not a human cell and that it is a pathogen cell, and that therefore there is something extremely wrong. Okay, so pathogen-associated molecular patterns, many of them have receptors on the surface of macrophages and dendritic cells, the two antigen-presenting cells, and they will bind to their associated receptor. And I want to stress that all of these receptors are grouped under the umbrella term, pattern recognition receptors, but there is not, it's not as though that there's one pattern recognition receptor which recognizes all of the PAMPs. No, no, no. Each of the PAMPs will have its own pattern recognition receptor. And by the way, pattern recognition receptors are often abbreviated to PRRs for short. Okay, so the archetypal example of this is the pathogen-associated molecular pattern LPS, which stands for lipopolysaccharides. Let me just write that down. So this is lipopolysaccharide, and this basically is a lipid molecule that no human cell would ever in its wildest dreams put on its cell surface membrane. Lipopolysaccharide, that's good. But gram-negative bacteria have it all over them, basically. And these professional antigen-presenting cells have a pattern recognition receptor for LPS, and this is what's known as Toll-like receptor 4, TLR4. So this stands for Toll-like receptor 4. And when the LPS suddenly appears in the vicinity of this Toll-like receptor 4, it will bind to it, and that will then trigger activation of the dendritic cell or the macrophage. So this tells the dendritic cell or macrophage that there is a pathogen in the interstitial fluid. So when these dendritic cells or macrophages have been activated by pattern recognition receptors, they will then start to produce co-stimulatory molecules and put them on their cell surface membranes. So basically, this is a checkpoint. This is to make sure that this professional antigen-presenting cell actually is presenting an antigen that is associated with a pathogen. And if the professional antigen-presenting cell has detected, if this antigen is from a pathogen, then the pathogen will also have had pathogen-associated molecular patterns associated with it, which will have activated the pattern recognition receptors, and therefore the antigen-presenting cell will have the co-stimulatory molecules on it. So it's to make sure that it is not presenting a self-antigen to fragments to these naive CD4 positive T lymphocytes. Okay, so it's a checkpoint. It helps make sure that we don't initiate an immune response against autoantigens. Okay, so what are these co-stimulatory molecules? Well, one of them, which we'll have as this one here, is what is known as CD40. Okay, so I'll colour in CD40 in yellow, I think. So CD40 is here in yellow. Okay, and then the other one isn't actually set. It can be two different molecules. Okay, and confusingly, both of these two molecules that you can put there, and which do the same thing, they also have two names. So it looks as though there's four, but in fact there's only two. So it can either be B7.1, or you can put B7.2 there. Okay, and these two molecules both have different names. So B7.1 is also called CD80, and B7.2 is also called CD86. Okay, so when the dendritic cell or the macrophage has been activated, it will put on its surface this CD40 molecule, and then it can also put B7.1 and B7.2 on its surface, which are also called CD80 for B7.1 and CD86 for B7.2. And these will also bind to another, well, both of them bind to a certain receptor on the surface of the T lymphocyte and do the same job, so they're equivalent, basically. Which one actually does the activation is trivial. Okay, so on the surface of the T lymphocyte, then, you have receptors that are complementary for each of these. So the receptor for either B7.1 or B7.2 is a protein known as CD28, and the protein that's going to bind to CD40 on the dendritic cell or macrophage is going to be a protein known as CD40 ligand. Okay, so it's CD40L. So let me colour these in. So we'll have CD40L in red here, okay, and then we'll have CD28 in vivid purple. Okay, so here it is in vivid purple. Okay, now when the co-stimulatory molecules CD40 and either B7.1 or B7.2 bind to their receptors on the surface of the T lymphocyte, which are CD40 ligand in the case of CD40 and CD28 in the case of B7.1 or B7.2, that is going to deliver what is known as signal 2 to the T lymphocyte. Okay, so you're now going to get signal 2. And signal 1 and signal 2 delivered together are then going to cause a response that is going to lead to the activation of this T lymphocyte. And we'll continue this discussion in the next video.